All right, ladies and gents, welcome back. So we're going to finish our discussion on cell transport today, really talking about active transport, in which case we need energy. So what does that really mean? So when scientists define energy, we're talking about the capacity to perform some sort of work. And that work can happen really in a lot of forms. We can talk about this gentleman in the picture and how he's working to ride that bicycle, which means he needs to move his muscles and get his heart pumping and move blood throughout his body so that they can maintain oxygen. But we can also talk about energy at a cellular level. And it can be something as simple as moving things around. All right. Moving organelles around, moving molecules around, moving things in and out of my cells is really what the topic of discussion is here. And so when we talk about energy at the cellular level, your cells have an energy currency. All right, think of it literally like money. You've got to spend it to do things. And that molecule, remember, is called ATP, or adenosine triphosphate. And it really is what allows your cells to do all sorts of things. And it's the reason you need to eat. All right, your body breaks that down into sugars, and the sugars then get broken down via cellular respiration in your mitochondria to make this ATP. So, really, when we look at this, here's your ATP molecule. So, it's a ribose, and adenosine, and 3 phosphates. And it's really in those phosphates where all this energy is bound up. So, if I break off a phosphate by adding water, so it's a hydrolysis reaction, right? I'm going to break it into smaller pieces. I create this molecule called ADP, or adenosine diphosphate. So tri is 3, di is 2. That free phosphate also then gives me a bunch of energy. And I can use that phosphate plus that extra energy to go phosphorylate or add that phosphate to other molecules. And as a result, I can make those molecules do things. So I can spend the energy by taking off that phosphate, attaching it to something else, and making a protein chain shape or making a molecule move somewhere else in my cells. So when we look at ATP, it's for a whole host of types of work in my cells. It can be chemical. So maybe I've got a really, really slow chemical reaction. I've got no enzyme to help me speed it up. So I can add that ATP plus that phosphate, break it up, use that extra energy, and now make a product out of it. I can do it for mechanical work. So this is actually moving things around, things like molecules and organelles, and I need stuff to go from one side of the cell to the other, break down that ATP, phosphorylate that protein, get it to change shape, and get it to move. We're going to focus more on this side of things for our purposes this unit, and that's that transport work. What if I need to move stuff from an area where it's low concentration to where it's already high? Go back to the tennis balls and dropping them in the middle of the classroom. Remember, we talked passive transport. If I drop that bucket of balls, it's where they're highly concentrated. Doesn't really take a whole lot of effort to just dump the bucket, and those balls are going to go from where they're already high in the bucket to where they're low, spread out across the classroom, and eventually they kind of stop moving. But if I need to go back in the other direction and clean up the mess, put those tennis balls back into that bucket, make them more concentrated, get them closer together, it's going to take energy to do it. And what's happening at my cells in order to do that for molecules like sugar or water, now I'm going to need ATP. I'm going to need to spend that energy to do it. So it's this big cycle. So if I look, here's where we're talking about on this side of the cycle, taking that ATP, removing that phosphate, getting energy as a result, and making ADP from it. When we talk about things like photosynthesis and cellular respiration, it's the other half of this. How do I take ADP, which I can't really use? It's like having a bunch of pennies that don't really buy anything. So I need to convert it into $20 bills. I need to convert it into something that's actually useful that I can spend now and actually get something out of. So it's a big cycle. Okay, and your metabolism is this side of things. Your cells doing work is on this side. All right, so when we take a look at what's going on in your cells and talk about active transport itself, here your cells are expending that energy, spending that ATP, literally like money, literally like currency in order to do something. And in this case, what it's trying to do is move those molecules from an area where they're already low in concentration and making them even more concentrated on the opposite side of the membrane. So go back to those tennis balls and I drop them on the floor, they all spread out. That's the passive side of things. I didn't need energy to do that. And those molecules, or those tennis balls in that case, are just going to keep spreading out until they're no longer running into something else. All right. But if I need to actually put them where they're more concentrated again, get them closer together, that means I have to expend energy to do that. Now, in order for your cells to do this, they're moving these molecules, whether it's sugars, whether it's water, whether it's salts, whatever the case might be. And in order for them to move against that concentration gradient, in order to spend that energy, it's going to have to involve a protein. So it really doesn't matter whether or not these solute particles are hydrophilic or hydrophobic. In order to expend the energy, I need the protein to move it. It's like having a pump. All right, so just like a water pump in your basement, some pump in your basement, you've got water in that basement where it's at a higher concentration, you don't want it there. That means you're going to have to expend energy, in this case in the form of electricity, to pump it out of your basement to somewhere that it's a lower concentration to get it out. Now, when we look at active transport, we're going to talk about a couple of specific types, but in general, you've got 4 basic steps to this. So here's step 1, right? Here's my transport protein. It sits in my phospholipid bilayer. I've got high concentrations of this solute on the outside, low on the inside. And the first step is just to get that solute to bind to the protein, to stick to it, so that that pump can actually hang on to it and move it. So that's this piece here, just that binding piece. Second step, here's the breakdown of the ATP. So 3 triphosphates, break one of those phosphates off, I get ADP, kind of just goes into your cells. We're going to make that back into ATP somewhere else later. And I'm going to take that phosphate and that extra energy, and I'm going to add it now to the protein. And by plugging this in, it's kind of like plugging in a pump into an outlet. You've activated it, you've turned it on, you've spent that energy, and now that's going to cause that protein to change shape. And what that does then is release your solute to the other side of the membrane where it's already high. That detaching of that solute then causes the protein to go back the other way, so sort of like kicking off a circuit, turning it off, breaking the connection. That phosphate detaches, that's the dephosphorylation piece, all right, and the protein goes back to the way it's sort of naturally configured, and that's what's going on over here. It's going back to its natural shape, so that the whole cycle can start all over again. So if we look at a specific pump, a specific type of active transport that's pretty common in your cells, we can talk about something like your sodium-potassium pump. And this particular pump is really important when we talk about your nerve cells. In order for your nerve cells to send a signal from your brain to, say, your muscles to get your body to move or do something, we have to change electrical impulses. We have to cause electricity to go across the membrane. But the only way to do that is to set up an electrical potential, set up charges on either side of the membrane so that that electricity can flow. So how does the sodium-potassium pump work? Well, it's got 6 basic steps. So if I take a look, the first step, if we come back here to the diagram, is this one right here. So that sodium-potassium pump is a transport protein. It sits in your cell membrane, and it's naturally open to the inside of your cell. So your sodium concentration is naturally low inside your cells. Now remember, this is active transport, so I'm going to move that sodium from where it's already low to where it's already high. So low inside, I'm going to add 3 potassium ions. Okay, those potassiums are going to, those sodiums, Na+. Those Na ions are going to get added to that protein. And in comes the ATP. So we're going to spend a molecule of ATP to move these sodium ions outside. So once that phosphate is attached, we use that extra energy, change the shape, and out go the sodiums. They're already high out here. Now we're making them even higher in concentration. I'm adding even more to it. As long as that phosphate is attached, it's going to remain open to the outside, and that's where this comes in. So now I'm going to take potassium, K+. Potassium's already low on the outside, but high on the inside. Add those potassiums to that protein. By binding them, that's going to kick off this phosphate group, cause that protein to change its shape back to being open to the inside of the cell. Those potassium ions then get released, and we're back to that natural configuration, open to the inside, add the sodiums, add the phosphate group, release the sodium, add the potassium, remove the phosphate, release the potassium, and the cycle starts over again. So by ensuring that there's high sodium outside and low potassium inside, that's going to allow us to set up this electrical potential. And when your cells actually send a signal, they actually flip those concentrations. And then we get ready for the next signal to come down. We got to reset the system again. So it's sort of like plugging something in, getting it to do something. It trips the breaker in your circuit box, right? And you've got to go reset it. That's kind of how this is working in terms of allowing those electrical impulses to continue to get passed down your nerve cells. Now, go back to what we started talking about way back when we started this unit, and we discussed what sort of things can go inside your cells versus what sort of things cannot pass across that cell membrane. And we talked about the hydrophilic, hydrophobic, polar, nonpolar idea, but size also was something else that we discussed. So what if instead of having to worry about moving stuff across the membrane that's small, what if it's big things like proteins or really big starch molecules? Well, then it's not going to matter whether or not it is able to interact with those fatty acid tails. It's not going to matter whether it's polar or nonpolar. Instead, now I've got to deal with size. And anything that's big isn't going to be able to fit through those little intricate spaces between those phospholipids. And so I'm going to have to do what we talk about as bulk transport. And that's where this idea of endocytosis versus exocytosis comes in. So exocytosis, like exit, all right, these are big molecules that your cells are producing, and they need to get them outside of the cell because they need to get used somewhere else in the body. But they can't move these big molecules across that membrane. They're just too big. So what ends up being produced instead is a vesicle full of those molecules, whether it's waste products or whether it's some sort of product that the cell is producing that needs to get used somewhere else. Think of like hormones or other proteins. And so that vesicle comes, it fuses with the cell membrane, and it spews those materials outside of the cell. All right, that's the idea of exocytosis. Why is it considered part of ATP, or active transport, I should say? It's because I'm going to need ATP to move those vesicles. So not really about a concentration gradient thing. It's just in order to move them, I need the energy to move the vesicles. And if you remember back to that endomembrane system we talked about in your cell structure, right? We talked about the nucleus containing the instructions. We went to the ER to build the proteins because that's where the ribosomes were. Those vesicles went to the Golgi to be your packaging, sorting, distribution center. And then the Golgi also made vesicles. Well, here's where those vesicles end up. So if your cells are building something like a hormone, hormones are proteins, so we follow that same progression. Nucleus, rough ER, vesicle, Golgi, vesicle. And in order for your cells to get that material out of the cell, to export it, to excrete it, or secrete it, depending on what kind of terminology you may come across, we need to get those vesicles to the cell membrane so that we can get those big molecules outside. And that's the idea of exocytosis. Endocytosis goes the opposite direction. So we're entering the cell as opposed to exiting it. But it's still big molecules. It still needs to bring those things inside. Still not necessarily about concentration gradients, but I need the ATP to get the vesicle formed and move it in. So here's all of these molecules in my environment. I need to take them in. So I form that vesicle, pinch it off, and now I can take this off into my cells. And a lot of times these vesicles or vacuoles are those food vacuoles, right? These are nutrients out here that your cells need to bring in so that your cell can do something. So eventually that food vacuole will go merge with a lysosome, break them down into smaller components, and then your cells can actually use them to make energy to do other cellular processes that they need to take into consideration.